I want to take you now 23 years ago. I was working on this very friendly paper in theoretical physics, but something really extraordinary was happening. I was carrying my first child, Yarden. And as I was doing that, a question kept running in my head. How can a single cell, the egg, can give rise to an entire human being? Have you ever stopped to think about it? Well, I couldn't stop thinking about it. And I had to leave physics and switch jobs, and I went to Caltech, joined the lab of Professor Davidson to learn about sea urchin development. Sea urchin. You ask yourself, what is the relationship between, she was talking about human embryos. Why is she talking now about sea urchin all of a sudden? Well, one of these movies is sea urchin development, and the other is human development. Can you tell which one is which? No, it's hard, right? Because the beginning is very similar. But unlike human embryo, we can do experiments in sea urchin embryos. And we can ask basic questions, like, how do cells make decisions? What do I mean by that? Okay, this is a sea urchin embryo, 4 hours post-fertilization. Every circle here is a cell. And these 2 cells over here already know at this stage that they are going to build this skeleton, these calcite spicules, 3 days later. How do they know? The mother tells them. How does she do it? She inserts a specific protein to the bottom of the eggs. So the cells that inherit this protein activate this network that is as enticing as my paper, I know, but let me walk you through it. Each node in this network is a gene. And an arrow going out of the node indicates that this gene encodes a protein that can activate other genes. So these guys over here are the decision makers. They tell each other and the other gene what to do, like our president. And the one over there are the builders, the genes that will actually build the skeleton. So, sorry, I lost the thing. So you already know it's not like a skeleton. I was supposed to ask you, do you think it's similar? No. Skeleton, human skeleton and sea urchin skeleton, 2 different programs. But in my lab, we discovered that the same managers that produced the sea urchin skeleton are the ones that drive our blood vessels formation. And what is the connection, again, between spicules, calcite spicules, and blood vessels? The answer is that both are biological tubes. In our blood vessel, the tube carries blood, and in the sea urchin skeleton, the blood inside the tube, there is a calcite, the mineral. Evolution works in a mysterious way, and you will hear about it in the next talk too. So what we do now in my lab, we take these skeletogenic cells, put them in a dish where they can grow these tubes without the entire embryos. They can do it themselves. And we study how they do it. And why do we care? Why do we care so much about biological tubes? Well, the same mechanism that drive embryo development underlie human diseases. For example, this is, don't be confused by this appearance. This is a tumor. And a tumor cannot grow more than a millimeter in size without the supply of nutrient and oxygen. And how does it get nutrients and oxygen? He summons blood vessels. He summons blood vessels that gives it oxygen and nutrient, enable it to grow and to metastase. So if we understand how blood vessels are made, we can block them. And if we can block blood vessels, we can block cancer. And this is one example of the biological tubes in our body that if we understand how they are built, we can cure diseases. So my name is Smadar דה-לאון. I told you in the end. I'm the head of the Department of Marine Biology. The nice illustration were made by my daughter, Yarden, that was the one I was carrying. And you're welcome to, if you want to hear more about it, and even better, if you want to collaborate with me, please go to my lab website or write to me. Thank you.